Okay, so our next speaker this morning is Ali Ellebedy from Washington University School of Medicine in St. Louis. Sorry,
okay.
It's remote. Okay, so we're looking forward to your talk.
Oh, thank you. I'm not sure if can you hear me well?
Yes.
Okay, great. Because the video is not working, and yeah, it's okay. You're not missing much. It's okay. So you'll just hear me, and well, I'll start by thanking really the organizers for the invitation. I'm very disappointed that I couldn't make it in person because of this some annoying virus, but I'm in the meantime I was really looking forward to first to interact with the community. And really to get introduced to this wonderful community. I've been working with Steve Kleinstein and Corey Watson, and I've had really great experience with everyone that belonged to this community. I was really hoping to meet you in person, and hopefully next time. So I will start by when I saw that I'm giving a talk after Shane, I had mixed feelings, because first I know that Shane always gives excellent talks, so it will be always a tough act to follow. But also I thought on the plus side, probably I don't need to do any introduction on B-cell or immunity to vaccines, because that's probably covered very enough before I start. But also there's one thing also, Shane was at least indirectly involved in the story that I'm going, in the beginning of the story that I'm going to discuss. So I'll go ahead and start by telling you about my disclosures, and they are not really related to any of the work I'll be presenting today. So first, the story started when I went to do a postdoc fellowship in Rafi Ahmed's lab in Emory. This was 2011, toward the end of 2011. And then that's a time when a couple of years earlier, a very classic paper came from Rafi's lab about studying B-cell responses in humans after seasonal influenza vaccination. And at the time, and remains till now, that really looking in blood is the main way, or the main technique for looking at immune responses in humans, B cells, T cells, or any other kinds of immune response. So in a very simple trial, healthy volunteers were recruited to the Emory Vaccine Center. The influenza vaccine was giving the seasonal vaccine, which is recommended every year for all adults. And then blood was taken before vaccination, 7 days, 2 weeks, and then a month later. And that's, as I said, very simple study. What was observed then is that you have at day 7 in blood, there's this population of B cells that gets expanded very quickly, and then also very quickly disappears from blood. And those turned out to be the Plasmablast. Those are the cells that are responsible for secreting the early wave of protective antibodies. And in the context of influenza, and those are the cells that their frequency correlate very nicely with the increase in antibody, serum antibody response from 0 to 4 weeks after vaccination. So they are responsible for this early wave. And from this study, we knew that in the context of influenza in humans, those cells are mostly coming from memory B cells, because they came quickly. As you can see, the rapid expansion, they were mostly isotype switched, and they already accumulated a high number of mutations. So they are really coming from affinity matured memory cells. So as I said, I started in his lab in 2011, 2012. And the main question that I was interested in is really what other immune cells being induced other than those terminally differentiated plasmablasts that we know they are not going to be lasting for long. And I use CD71 as a proliferation marker, a surface proliferation marker, to identify those cells that are also responding to vaccination, coming around the same time as plasmablasts, although they are peaking maybe at 2 weeks instead of 1 week. This is all in blood. And those are the cells that eventually we propose that they become resting memory cells. This work was done in collaboration with Scott Boyd's lab at the time, Stanford. And at the end of this, my fellowship, the question that I became very obvious and very important that I wanted to address is, do we actually induce any germinal center response in this vaccination? This vaccination is being given to 150, 160 million people in the US every year. And we don't know. We know it's a suboptimal vaccine for many reasons, but we don't know really whether this is actually induces a germinal center. And of course, you know from Shane work and others that the importance of germinal center response, especially in terms of inducing long-lived immunity, plasma cells and memory cells. So how to study germinal center response? And this is around the time that the papers were coming out from Shane's lab for the using of fine needle aspiration in non-human primate. And I was starting my lab in 2017 in WashU, and this is the time where I thought, can we actually adopt this technique to actually look at influenza response in humans? And to make a long story short, during my first year at WashU, I worked with ultrasound radiologists, and I said, this is a diagnostic technique that you use every day with patients with suspected lymphomas or breast cancer metastasis diagnostics. So there are many ways we can many reasons to sample axillary lymph nodes. But I thought, can we actually sample that after intramuscular vaccination? And those will be the nodes or the cluster of the nodes. There are different clusters of axillary lymph nodes in humans. It took us the first year to learn which nodes to actually sample that are likely going to be the draining node. And I'll mention there are different clusters of nodes here in the axillary region in humans that have nothing to do with the deltoid muscle drainage. So they don't really, sampling those will not be helpful. So the way we sample, as Shane mentioned, we try to sample the cortical region, and we basically copied the technique from what has been proposed in NHPs, and we did it in our human participants before and after vaccination. And slowly we became good at predicting which nodes are likely going to be responding, so we can actually sample it before vaccination. And this is an example for that. Excuse me. I'll be coughing maybe a little bit, not just, I'm actually tested negative. It just, when I talk too much, there's this residual cough that comes. So excuse me for that. So before vaccination, you'll see that the node is really, the cortex of the node is very thin. And one thing I forgot to mention, that we track the same node over time, and that allows us to really see what happened, if any change happened in that node. So as you can see, before vaccination, based on the location of the node and everything, we learned that this is likely going to be draining. So you can see that 1 week after vaccination, you can start seeing thickening of the cortex. And if you now sample the B cells or analyze the B cells that are coming out of this, obviously in blood, you can see the plasmablast early on, but in the draining node here in the B cells, you see at 2 weeks, you start seeing the germinal center starting to emerge. And then those cells are responding to vaccination because they, at least impartially, bind to HA probes. And then one thing, once you have those cells, you now can answer, we answer a very important question that I mentioned in Rafi's first paper. We think plasmablasts are coming mainly from pre-existing memory B cells. So what are the cells composing the germinal center response in the same individual? So to do that, we single cell sorted total plasmablast from blood at week 1 and germinal center B cells at multiple time points from the lymph node. And then we made the corresponding monoclonal, cloning the heavier light chain, and then verified, obviously, the binding by ELISA for those monoclonals. So now we have the fingerprint of those responding B cell clones in the 2 different compartments. And then if we go back now and do single cell RNA-seq over time from the 2 compartments in blood and lymph node, and overlay the BCR data on those 2 compartments over time, we can actually now look at not only the level of mutations, which tells us the origin, potential origin of the responding cells, but also potentially the fate of the cell. Excuse me. So when you're looking at the B cell compartment in blood here in PBMCs, we are seeing that there's the 4 naive B cells, resting memory cells, activated cells. These are the cells that we worked on defining I worked as a postdoc fellow working on it. You can see plasmablast here, The same compartments, different proportions, but obviously in the lymph node here in the bottom, you see the germinal center. So I mentioned that we already made from those individuals the monoclonal antibodies, so we know the BCR information of antigen-specific clones. So now this is obviously total cells. If you overlay the antigen-specific, and you won't be able to see it here, very tiny. There are colored dots here. Those are antigen-specific clones, B-cell clones. Mostly at baseline, they're obviously in the memory compartment, either both blood or lymph node, very low frequency. Now at the first week, if you now overlay the BCR clones, obviously Plasmablast are, you can see those are antigen-specific. Not much happening in the draining lymph node at that early time point, at least in terms of antigen-specific responses within the area that we sampled. One thing that now we were able to do is we were able to assign the B cells if they're only detected in the plasmablast, we give them a red color. If they are detected both in the plasmablast and in the germinal center, they are in purple. But then if we detected them later or only in the germinal center compartment, but never in the plasmablast early on, we give them the light blue color here. And you can see that over time, you see that the activated B cells in blood increase in frequency, and that's we've really shown by flow. But now the germinal center, starting at 2 weeks, you start a more involvement in the germinal center in terms of antigen-specific response. And then you can see it up to, at least in what we have published so far, up to 2 months after this non-adjuvanted vaccine, which was a surprise at the time. So one thing is I mentioned is those cells that are only detected in the germinal center, we knew that the plasmablasts are coming from memory. But then we thought that those cells that only detected later are potentially coming from a naive compartment, that really novel B-cell clone being involved in this relatively prolonged germinal center with this non-adjuvanted vaccine. So to look at that, we worked with Steve Kleinstein lab at Yale, and we compared the level of maturation or level of mutations in those clones compared to the plasmablast in early blood. And this is published, so I'm going quickly to show that from 3 different individuals, this is the level of mutations in the heavy chain, and you'll see that the plasmablast only in red, shared in purple, and from the 3 individuals coming and later in the germinal center in blue, they are significantly less mutated. And work with Andrew Ward showed that those are structurally targeting novel epitopes in the HA. So the model that we thought about that we don't think those naive B cells are replacing the memory cells that are in the germinal center. The memory cells are continue to be there, but we think that having that extended germinal center provide a window for a window for naive B-cell clone to be recruited to the reaction in an immune response that traditionally or typically dominated by those we call high affinity memory B cells. So at that time, you know what happened, SARS-CoV-2 pandemic started, and we were not able to finish our work to look at the fate of those cells, either as a memory B cells or in the bone marrow compartment. So, but we were fortunate in a way that we can actually use that setup to address important questions with a novel vaccine platform in humans, which is mRNA vaccine. So the main question we wanted to address is, well, this is a novel vaccine. We wanted to see how robust the plasmablast response, and this has been addressed by many other groups as well, how robust and persistent the germinal center response to this vaccine platform, and the fate of those cells. And this is when we added the new component to sampling the 3rd compartment, which is the bone marrow after vaccination. So the study is simple, relatively. We recruit healthy participants who are getting the vaccine. At that time, obviously, this vaccine was at Pfizer at the beginning. 42 individuals, 7 of them had confirmed infection months prior to enrollment. And then we sampled blood and lymph node before and after vaccination, 1st and 2nd. At the end of the 6th month, we sampled the bone marrow from willing participants, obviously. And the plasmablast response is as expected. After first immunization, you see spike-binding IgG cells, and this is a very simple ELISpot assay to look at the IgG binding to the spike frequency-wise in blood. 1 week after 2nd vaccination increase, and then they disappear. And you can see this in antibody response. So if you actually now look at the Germinal Center, this is our best participants in influenza vaccination. You look at the by BCL6 staining, this is an intracellular staining that really, those are the typical Germinal Center B cells. And this is gated on isotype switch B cells. So this is all B cells and naive B cells are taken out. 38 intermediate BCL6 positive, those are Germinal Center. We were very happy to see this because you don't get a really robust response to, especially in the Germinal Center, to this vaccine. If you look at the same population in the first 4 participants in the Pfizer trial, this is the same population, and that gives you why people were having some enlarged lymph node in a way, because we have a really massive, very robust reaction. We've seen antigen-specific Germinal Center reaction every single person we looked at that received an RNA vaccine, and I actually can remember the number now. We definitely crossed 30 or 40 people, or closer to 50 actually, with any different kinds of mRNA vaccine. We always see antigen-specific response, which is something we don't see it for influenza vaccination. But then, as Shane mentioned, one of the things that we were curious about is how long that Germinal Center can persist. So as I mentioned, we track the same lymph node, and we track them for the 6 months after second vaccination. And 10 out of the 15 individuals that continued all the way to 6 months after second vaccination, we saw that not only a robust Germinal Center that is mostly spike specific, as you can hear. So this is gated on Germinal Center B cells, spike binding, and that's just looking at the frequency of those total GCs or spike-specific GCs out of all CD19. So you will see that for most of them, it's basically a flat line in those lymph nodes. So this was surprising for us to show that this is obviously shown first time in humans, that you can actually have the Germinal Center that at least going for 6 months. We actually track them all the way to 9 months. We haven't published this data yet, and at the time they started getting their booster immunization, so everything changed. But for those participants to have a vaccine for 6+ months, having Germinal Center ongoing, that was remarkable. Well, another thing that we found that we didn't really appreciate before is this population of long-lived, or not long-lived, lymph node plasma cells. Those are plasma. I talked about Plasmablast as antibody secreting compartment very early on, but we also know of the bone marrow plasma cells, and those are the cells that come at the end of come toward the end of the Germinal Center reaction to actually become our long-term compartment in the bone marrow. But we started seeing in the lymph node at around the same time seeing Germinal Center, we see this lymph node compartment, and we define it as, again, isotype switch B cells, CD38 high, so not intermediate. And obviously they downregulate CD20, but also to confirm it, we see that they are PLIMP1 positive. The antigen-specific staining is not as robust as the Germinal Center, but definitely there. And we can see them the same time we see a Germinal Center, so we can see them for the entire 6 months after second vaccination. But then the main question that really are those cells seeding our long-lived plasma cell compartment in the bone marrow. So we had 11 participants that actually gave us a bone marrow aspirate at the end of this, at the end of the 6 months. So I'm going to show you in the next few slides that we think that those bone marrow plasma cells are indeed coming from the lymph node plasma cells, but how we actually assess the bone marrow plasma cell compartment. So we take bone marrow aspirates, we enrich for CD138 positive cells, and this is very important detail because those cell frequency in the bone marrow aspirate is very low. So to be able to assay them, we just put them on because they are spontaneously secreting antibodies, we can actually just put them on plates that are coated with whatever antigen of interest. And if they are producing antibodies, every spot here will be one corresponding to one cell. So this is showing that against 3 different antigens, so against influenza, and this is obviously influenza vaccine from that year, that's a year ago, and then the tetanus diphtheria antigen is a commonly used vaccine that everyone has some history of exposure. more or less, and of the SARS-CoV-2 spike. And as you can immediately see here, everyone in our participant, and that's not really a surprise, had a lot of history of exposure to influenza antigens, and that led to accumulation of this large number of large frequency of plasma cells in their bone marrow. It's almost 1 in each 100 plasma cell in those participants. But then a much lower came the SARS-CoV-2 spike. So this is at the end of 6 months of a persistent Germinal Center. And as Shane mentioned, we can obviously, all of us saw that if you haven't been exposed before in a naive and you got the 2 doses of the RNA vaccine, we do see this decay over time till you get the 6 month. The fact that we still detectable antibodies at that time, long after the Plasmablast, assuming we died and they are no longer contributing to that, then we think that this is coming from the long-lived plasma cell. But if this is the case, those antibodies secreted at the time must be of higher quality than those that secreted early on, because those are the source of those cells should be affinity mature plasma cell or lymph node plasma cells. So to address that directly, we went back in a way that's really agnostic to the responding cells. We looked at we didn't use any probe to sort the cells. We just single cell sequenced from 8 different individuals, all the germinal center and lymph node cells, B cells, and then we did a clonal hierarchy of those cells, and any clone that was represented by 3 or more cells, we expressed the corresponding monoclonals. And that way we hope to have a good representation of the responding B cells in those individuals, at least in the germinal center compartment. And then verify that those B cell receptors or B cells are antigen-specific by testing the corresponding monoclonal antibodies. And we end up, after revisions, we end up doing close to 2,500, more actually than 2,500 cell monoclonal antibody expressed. 1,540 of them were antigen-specific. So now we have for these 8 individuals, those different clonotypes that are antigen-specific that we can go back and track the evolution of the cells. So the first
4
question,
minutes.
the first, oh, sorry.
Sorry, 4 minutes time left.
Okay, I will. Well, I think I have 5 slides. So the first part left. The first question asked is how different is the Plasmablast from the Germinal Center, and clonally they can actually be partially overlapping in those 8 individuals. But the fact that we can see them and we can see Germinal Center at different time points, those spike-specific clones, we looked at their level of maturation, and as you can see, early on, week 4, you can see equivalent level of maturation, but over time, you see higher maturation in the Germinal Center. So there's obviously an accumulation of mutations in the clones that are being persistent in the Germinal Center. If you now do the same comparison between lymph node plasma cells and Germinal Center, they're basically equivalent, overlapping clones 100%, and the mutations are accumulating at the same rate. Now, if you do that with the bone marrow plasma cells, what we see is that now if you compare Plasmablast, this is taking 1 week after second immunization, this is 6 months, and then within the 6 months, you sample the lymph node plasma cells. What we see that the level of maturation is actually increasing nicely over the 6 month period, ending up being those highest level of maturation of mutations are those in the cells, in the spike-specific cells we see in the bone marrow plasmas. So as I mentioned, those antibodies are likely being produced by this population of cells. And so they should be of higher quality in terms of avidity binding compared to those cells produced early on. And this is indeed the case by a very simple avidity ELISA, that the same individual, if you take their polyclonal spike binding, very low avidity at the beginning, that is significantly increased over 6 months because of this persistent Germinal Center reaction. And just to finish this off, we tracked individual clones over time. So this is a clone this is the same clonal lineage from a Plasmablast isolated very early on in blood. And then the same clone over time as a lymph node plasma cell or as a bone marrow plasma. And then you compare their neutralization, you see that they actually become better neutralizers, the same clone after it accumulates mutation that lead to their enhanced binding affinity. And this time we're looking at monoclonal, so this is binding affinity. So we think that now this accumulation of mutations in this persistent germinal center reaction after 2 doses of Pfizer not only led to having a population of bone marrow plasma cells that high accumulated or affinity matured, but also having a really a novel subset of cells that we haven't really been able to track before. We think they are a transitional stage before being seeding the bone marrow. And obviously we have also high affinity memory cells that if for a third immunization, they now respond robustly. And just to give you an example of the level of maturation, they always have, so this is what we started was after 1 week after second vaccination. Now, if you look at the same Plasmablast 1 week after third immunization, this is the level of maturation. So they are obviously coming from those matured memory B cells. So this was my last slide. Those are the individuals that are who really did the work. Jackson and Usab, they are postdoc fellows in the lab who actually did all the influenza studies and the COVID studies. Julian is an incredible scientist who joined me from Stephen Kleinstein lab, and he did all the clonal analysis, clonal tracking, and all of the mutation data. Obviously, very grateful to my clinical colleagues who actually, without them, actually, I wouldn't be able to do all of these really unique studies. A lot of collaboration at St. Jude to do the T-cell analyses that I then present here. Obviously, long-term collaboration with Steve and his lab been very, very important for my lab to start and for many, many things that I couldn't really do. Grateful for funding and obviously for volunteers. I'm happy to take any questions.
Yeah, so thank you, Ali, for this great presentation. So the talk is open for discussion.
Hi, Ali. Steve Boster, Emory University. We miss you up there in Chicago. Beautiful work. I was wondering if you could comment a little bit more on the phenotype of the lymph node plasma cells relative to blood plasmablasts. Can you see antigen on the cells with a bait, those sort of things?
Oh, yes. Actually, we can, and that's actually what I'm showing it here. You can actually see that the binding to the antigen. It's actually interesting. If you try to cluster, if you just try unsupervised clustering of the 10x data from the Germinal Center, and if you add bone marrow plasma cell to the mix, those cells actually cluster more with the Germinal Center. They have a lot of Germinal Center genes remained on them, in a way, if I can say that. Many things, many expression of genes that are very related to antigen presentation, recent proliferation, and many other things that you can definitely tell, oh, I know this is expressed on a typical Germinal Center. Obviously, they don't have BCL6 and the upregulate LIMP1 and many of the other things that we know important for antibody secretion and plasma cell differentiation. So I think they are, I think they are in their way. Which ones make it to the bone marrow? We have no idea.
Ali, it's Shane. That was great stuff, and you know how much I like all of this. And also, you sounded really clear on this whole talk, by the way. It definitely came through fine. We heard, well, yeah. Anyways, I'm also curious about the phenotypes of the lymph node plasma cells. You mentioned that many of them had signatures of recent proliferation. Was there any subpopulation that looks like it's going to become a resident lymph node, potentially long-lived plasma cell? I mean, Stuart Tanguy and others have seen in spleen, right, that bone marrow is not the only place you could have long-lived plasma cells, that you could have them in other lymphoid compartments. But I don't know how much evidence there's been for a real lymph node resident population. Did you see any evidence of that, or were they really just looking like transient residents?
Thanks, Shane. Great question. And there's a paper also came in 2000, I think it was 2007 or 8 from Mark Slifka's group that showing there is a lymph node in the lymph node plasma cell in humans. Oh, I think it was NHPs actually. Yeah. So in that case, in the 5 individuals where we actually didn't see at the 6-month time point, didn't see any evidence for Germinal Center specific or spike binding cells, I thought we will be, if those guys are resident, we will see them there because they don't have to have the Germinal Center ongoing. But for us till now, every time we see them, there is always a spike specific Germinal Center at the same time. If those are gone, those are gone. So it's not a definitive, but in my mind, I don't see any sign of residence in those cells yet.
Thanks, Ali.
Hi, Ali. Question from Donald Lee at Eli Lilly. Can pre-existing non-neutralizing memory B cells interfere with the selection of new neutralizing B cells in germinal centers?
Yeah, beautiful question. We haven't, I can think of different ways. We haven't addressed that in this study. We are trying to address it in a different, and it's almost impossible, like to address that definitively in any human study, actually. We are trying to address it in a different model, but I can think of different ways. Yes, you can interfere by, even if those epitopes are not overlapping physically. If they are physically overlapping, obviously the interference is, will be direct. But if they are not even overlapping directly and they are different parts of the spike or the HA or whatever it is, I think the simplest way to think about that, you can interfere by clearing the antigen and making it less available to have a persistent germinal center, for example. And that will be an interference. I can think of different other scenarios as well that this could be could really affect the responses indirectly. But also I can think of the other way, having those antibodies or having these immune complexes that binding to this non-neutralizing could actually, have that immune complex in the germinal center could actually provide a better presentation in a way, if you can, of those neutralizing B cell clones inside the germinal center. I think this part is a bit fuzzy in the field, and it will be nice to have more clarity on what those antibodies can provide. Obviously, we learned a lot from the HIV field in that case about the impact of non-important epitopes and interfering with broadly neutralizing ones.
Okay, last question, Dan.
Yeah,
short question, short answer, please.
Thanks, excellent talk. This is just a twist on that last question about competition, but it's about the plasmablasts, and do the early time point plasmablasts, which are not efficacious per your data, they're secreting antibodies in blood that can be competing against the memory B that are more efficacious. And is that basically a tension between, actually developing an efficacious response in the long run and these short-lived cells that can confound it?
Okay, I love this question. It's very hard to answer it without really being in a long. So please email me because I think about this question a lot. So I can tell you the short answer is I think yes. They're potentially not only affecting it, potentially directing the response in different ways. And we are trying to directly assess that. So I'll just keep it short. But one thing that I should note that I did not show that those antibodies are not efficacious. I showed that they are just having lower avidity, but it doesn't mean that they couldn't be protected. Okay.
I'll follow up in email. Thanks.
Thank you.
Okay, thanks again.